Hi, this is Dental Basics, and I'm Dr. Parvati Raghavan. Today's topic is about nerve impulse generation and conduction. Let us first take a look at the different terms explained throughout this video. They are stimulus, impulse, action potential, resting potential, polarization and depolarization, saltatory conduction, lipid bilayer, polar and nonpolar, membrane proteins, hydrophilic and hydrophobic. The structural unit of the nervous system is the nerve cell. This is the cell body with many dendrites arising from it. Below is a single axon. An axon can be myelinated or non-myelinated. When myelinated, it is covered by the myelin sheath. There are gaps in between the myelin sheath, and these are called the nodes of Ranvier. In the end of the axon are the axon terminals. When we compare the structure of the nerve cell and other cell bodies, similarities are that all are surrounded by cell membrane, contain cytoplasm, mitochondria, and other organelles. Difference is that nerve cells have an axonal process at a distance from the cell body. This is an axon with myelin sheath. The myelin sheath is made of lipoproteins, of which 75% are lipids, 20% proteins, and 5% carbohydrates. Nodes of Ranvier are gaps in the sheath at regular intervals. Impulses jump over the myelin sheath to the nodes of Ranvier and travel much faster as compared to unmyelinated nerves. If you look at the image closely, you can see that at these nodes, the nerve membrane is directly in contact with the extracellular fluid. The cytoplasm of the nerve is called the axoplasm. The cell membrane of the nerve, that is the nerve membrane, is called the axolemma. Here you can see a cross-section of the nerve, axoplasm, axolemma, and extracellular fluid. Axolemma in some nerves is covered by myelin. The axoplasm is different in the dendrites, cell body, and axon. The difference is in the composition of organelles and other materials inside. The composition of the membrane is also different in different parts of a single nerve. Membrane in the axon is specialized for electrical conduction. Membrane at the end of the nerve is specialized for synapsing. All the specialization helps to perform different functions. The central nervous system is made of two types of neurons: motor neurons, connected to muscles, and sensory neuron, which conducts sensation of sight, smell, taste, touch, etc. Both motor and sensory nerves have different structure. Also, one receives message from the CNS while the other delivers messages to it. Let us know a little bit more about these two neurons. The motor neuron receives information from the CNS. This is a motor neuron, dendrite, cell body, and axon. The cell body is present between the dendrite and the axon. It is involved in impulse transmission and provides metabolic support, that is, energy for the entire neuron. The sensory neuron carries information to the CNS. This is a sensory neuron, dendrite, cell body on top, axon, and axon terminals in the CNS. Dendrites respond to stimulus produced by the tissue and generate an impulse. Axon can be very long and functions to transmit impulses. The sensory nerve cell body has one main function, that is to provide metabolic support, that is energy, for the entire neuron. If you look at the image, you can see that it is at a distance from the axon. It is not involved in impulse conduction. Nerve functions to carry messages from one part of the body to another. These messages are in the form of impulses. Impulse in English means a change that happens suddenly. The nerve impulses are also sudden. Impulses always travel in one direction from the dendrites to the axon terminals. Why? The answer is in part 2. Each axon terminal then forms connections with many other neurons across tiny gaps called synapses. The impulses on reaching the axon terminals trigger release of chemical substances called neurotransmitters, which pass through the synapse, causing the connecting nerve to catch the impulse. Stimulus causes generation of an impulse. Let us find out what a stimulus is. Free nerve endings, that is dendrites, have receptors that lie in the tissue. These receptors are sensory and respond to stimuli, which are basically changes in the tissue, which may be chemical changes, mechanical, change in light, or temperature. So these receptors are accordingly called as chemoreceptors, mechanoreceptors, photoreceptors, and thermoreceptors. And these initiate the impulses. This is an unmyelinated axon. You're going to take a cross section of it. This is the axoplasm in the middle, the nerve membrane. Take a section of this and enlarge it. Nerve membrane, axoplasm here. And extracellular fluid outside the nerve. The membrane is covered on both the inner and outer surfaces by a single layer of non-lipids. In the center are the bilayer of lipids with polar heads. Next, let us look at the bilayer of lipids. In living things, many biological molecules like proteins and DNA are formed based on a stable type of bond called the covalent bond. In this bond, two atoms share electron pairs. By sharing their outermost or valence electrons, atoms gain stability. This form of sharing can be unequal or equal. When unequal, they're called polar molecules, and when equal, they're called as non-polar molecules. The lipid heads in the nerve membrane are polar and point outwards, and the lipids are phospholipids. The phospholipids are made up of two parts, head and tail. The head consists of a phosphate group and a glycerol molecule. The tail is made up of two chains of fatty acids. This is a close-up of the phospholipid bilayer, head group and tail group. The head group is polar and the tail group is non-polar. The phosphate group in the head can be modified with the glycerol, choline, ethanolamine, etc. Biological membranes are fluid because these hydrophobic tails prevent the heads from packing together and forming a solid. The chemical formula of the phosphate group shows that it is negatively charged. Each phosphate group contains one negatively charged oxygen atom which gets attracted to water in the axoplasm as well as the ECF and forms hydrogen bonds. It is hydrophilic. And because of this, the phospholipid spontaneously forms bilayer in water with the head group facing out and the tail group facing in. The fatty acid tail consists of 14 to 24 carbon chains. They are uncharged and hydrophobic. This is the lipid bilayer. 1, 2, 3, and 4 are the membrane proteins. These membrane proteins can be integral or peripheral. Integral proteins are integrated and permanently anchored to the membrane. They are anchored to the membrane core, which is hydrophobic. So it is necessary for them to have at least one hydrophobic region. In the image, you can see that protein 1 sticks only partway into the membrane, while protein 2 stretches from one side of the membrane to the other. Protein 2 are called transmembrane proteins and have hydrophobic areas present inside the membrane and hydrophilic areas facing outside. Three and 4 are peripheral proteins and are found on the outer and inner surfaces of the membrane. They are loosely attached by ionic bonds or calcium bridges to the electrically charged phosphoryl surfaces. A typical cell membrane generally consists of around 60% proteins. Why so much proteins? That is because they play a vital role in almost every process in a cell. Proteins function as catalysts, which we call enzymes, transporters, pumps, and ion channels, receptors for hormones, local mediators, neurotransmitters, energy transducers, etc. Energy transducers transform one form of energy to another, that is stimulus, into electrical potential difference across a biological membrane. More proteins are present in more active organelles like the mitochondria. Nerve membrane has selective permeability, which creates a difference in concentration of ions on either side of the nerve membrane. This makes the inside relatively more negative with a potential difference of -60 to -90 millivolts. It is called the resting potential of the nerve. Potassium is the main abundant cation in the ICF of all type of cells like muscle, skin, subcutaneous tissue, RBC, and also nerves. ICF has more potassium ions and ECF has more sodium ions. Sodium and potassium both are cations, that is positively charged. The question is then, how is the membrane positive on one side and negative on the other? This will be explained in part 2. This potential difference between the two sides of the nerve membrane leads to what is called as conduction. When the inside of the nerve membrane is negative, that is in resting phase, the neuron is said to be in a polarized state. A change may cause the inside of the nerve to be less negative or relatively positive to outside. Then the neuron is said to be depolarized. A neuron can also become hyperpolarized, and we will be reading about this in part 2. Now we are going to learn how impulse spreads in an unmyelinated nerve and a myelinated nerve. Take these 3 segments of this nerve. This pinkish color represents impulse in the first segment of the nerve. As the impulse spreads, it moves forward to segment 2 and then move forward to segment number 3. Thus, the spread of impulse is a forward creeping process from segment 1, then to 2 and to 3. In a myelinated nerve, the impulse leaps from node to node and is called saltatory conduction. Saltatory comes from the Latin word saltere, which means to hop or to leap. Impulse can be compared to lighting a fuse. Once lit, the fuse burns steadily, one burning segment providing the energy to ignite the adjacent segment. In the same way, once stimulated, the impulse has the same amplitude as it passes along the nerve length without losing quality or strength. The energy for this propagation is derived along its length. Thus, changes in the nerve membrane cause excitability and conductivity. Nerve membrane has selective permeability. It acts like a semi-permeable membrane and has electric potential, that is energy arising from collection of charges. All these features of the nerve membrane are going to be covered in detail in part 2. Before finishing off this video, I want to put an emphasis on 2 points that 1, intracellularly, the nerve cell is rich in potassium, and 2, it is negatively charged. Here's a sneak peek of the few topics in part 2. Different types of diffusion, formation of electric fields across the nerve membrane, the sodium-potassium pump, different ion channels present in the nerve membrane. Thank you for watching and sharing, and remember to press on the like button below and subscribe.